There are bound to be times when you want to linger on images in this presentation, so please make good use of your pause button. We have 3 Polysticum species in Britain and Ireland: Polysticum lonchitis, the holly fern, Polysticum aculeatum, the hard shield fern, and Polysticum cytiferum, the soft shield fern. First, we'll consider identification of those 3 species. Later, their hybrids. Here are fronds of the 3 species with examples of their growth habits. The hard and soft species may at first be difficult to differentiate, but keep going. You might note that those names already tell you something useful. The fronds are quite distinctly different, shown here twice. Of the 5 ways in which fern fronds are divided, only 2 concern us in the Polysticums: once divided or pinnate, and twice divided, bipinnate. At first, difficulties might be caused by superficial similarities with the fronds of several other ferns, such as the male ferns, lady fern, lemon-scented fern, and perhaps the marsh fern. But don't worry, you'll soon tell them apart. To facilitate identifications, I've produced 2 field guides. Identification Stage 1. Turn the frond over to examine the spore-producing organs, the sori, singular sorus. Each sorus consists of a heap of spore capsules, the sporangia, protected by an umbrella-like membranous cover known as the indusium, which helps us distinguish Polysticum from other fern genera. In all Polysticum species, the indusium is circular with a wavy margin, robust, though shrivelling, right, and persistent. By comparison, the indusia of male ferns are also circular, but with a segment missing, below. They're sort of kidney-shaped, also robust, rolling or shrivelling, and persistent. The sori of lemon-scented fern are quite different, circular with a small ragged indusium which usually shrivels and disappears early. The distribution of sori is different from all other British ferns, being strictly marginal, on the edge, hence limbo sperma. The sori of lady fern are half-moon or J-shaped, with a particularly ragged indusium. Identifying the 3 Polysticums requires some attention to detail. The main segments, the pinnae, singular pinna, are undivided in the holly fern but divided in the 2 shield ferns. Note the pointed thumb-like projection on each holly fern pinna, or the pinnules of the shield ferns. It's characteristic of all Polysticum species. The fronds of both hard and soft shield ferns are subdivided, their pinnules characteristic of each. From here on, holly fern may be accepted or discarded. With holly fern excluded because of its simpler frond structure, we can now concentrate on the 2 bipinnate shield ferns. The fronds of the hard shield fern feel tough to the touch and the hairs are stiff, almost little spines. Many pinnules have a broad, flattened stalk, while those towards the pinna apex become progressively unstalked, fused to the costa. They are, as on all Polysticums, mitten-shaped, but the thumb is reduced compared with the soft shield fern. Its fronds are relatively soft to the touch, and the hairs are also soft. As well as having that much more prominent thumb, all pinnules are distinctly stalked. Here are some general points which help with initial identification. Holly fern is quite rare, found mostly in Scotland, and grows only in base-rich soils, on limestone and volcanic rocks such as basalt. Hard shield fern is common, growing pretty well everywhere in the UK, often where it has its roots in wet alkaline conditions on rock faces and sometimes on base-rich soils in woodland. Soft shield fern is also common, though almost absent from northern Scotland. It's usually found growing in base-rich woodland soils and on stream banks and waysides. Descriptions of the 3 species. First, the holly fern, here seen with the green spleenwort, another fern of calcareous soils. Its fronds are quite stiff and often upright. They're narrow and divided once, with tough, prickly, mitten-like pinnae, each with a prominent thumb-like basal lobe. The sori are typical of all the Polysticums. Holly fern occupies 2 rather specialised habitats, Here a basaltic mountainside on the Isle of Skye, where it grows in rock crevices and in soil pockets on ledges. Note that it is, as often in this sort of habitat, accompanied by roseroot. And here is its limestone habitat at Glasnock in Wester Ross. Here, another typical habitat-sharing plant is the lovely mountain avens, and sometimes even next, hardshield fern, Polysticum eculeatum, beginning with a sequence of pictures showing habitat and habit. While soft shield fern usually grows as a regular shuttlecock, hardshield fern fronds tend to arc over to one side, downhill and presenting maximum leaf area to the often limited light source. Even when the slope isn't particularly steep, fronds tend to arc in one direction. With hollyfern on limestone, their hybrid is not very far away. Shield fern fronds are quite distinct. The spread of the lowest pair of pinnae is characteristic, narrow in hard shield fern, broad in the soft. Soft shield fern, Polysticum certiferum. Its habitat is usually better drained than that of hard shield fern, woodland and wayside, but always on neutral to high pH soils. The fronds usually form a regular shuttlecock. Some frond details and pinnules close up. The spread of the basal pinnae is characteristic, broad in soft shield fern, narrow in the hard. At this point, I will introduce three pteridologists who worked in the science field known as cytogenetics, the study of inheritance in relation to the structure and function of chromosomes. Professor Irene Manton at the University of Leeds was a pioneer of the science, specialising in the ferns. Two of her students who applied her methodologies were Anne Sleep and the Hungarian Gabor Vida. All three contributed significantly to our understanding of the genus Polysticum, and it is their research which answers the questions posed by this presentation. All three of the Polysticum species found in Britain and Ireland form hybrids. We will now create a diagram which will be used throughout the hybrid descriptions that follow. It's important to note that two of the species are diploids with 82 chromosomes each, while the third, Polysticum eculeatum, has a double set. It is a tetraploid with 164 chromosomes. The existence of the tetraploid is very significant and will be discussed later. The three Polysticum species and their three hybrids. The triploid hybrid of P. eculeatum and P. certiferum, Polysticum bicknellii. The triploid hybrid of P. eculeatum and P. lonchitis, Polysticum illyricum. And the diploid hybrid of P. certiferum and P. lonchitis, Polysticum lonchitiforme. Always with reference to that scheme, we'll look at the three hybrids, beginning with the commonest, Polysticum bicknellii, the parents of which are very common, the hard shield fern, Polysticum eculeatum, and the soft shield fern, Polysticum certiferum. Polysticum hybrid bicknellii, P. eculeatum crossed with P. certiferum. Polysticum bicknellii is seen here in roadside woodland near the Hope Valley Reserve in Shropshire. It's an excellent site for pteridologists because the parents and hybrid, plus a lot of other ferns, are plentiful. Whole fronds of the parents, and then with the hybrid. Here are the hybrid's parents compared at the Hope Valley site, and here their hybrid, appearing exactly intermediate between them. Examination of the pinnules confirms the hybrid's intermediate morphology, not to mention its very evident expression of hybrid vigour. The spread of the basal pinnae is also intermediate. Back to the three hybrid diagram. Polysticum illyricum is worth spending time on because, although it is rare in Britain, maybe only 11 occurrences, it's also capable of forming a colony, as we shall see. That is not usual for hybrids. Polysticum hybrid illyricum, P. lonchitis crossed with P. eculeatum. It's a rare hybrid for which, fortunately, we have plenty of information. Polysticum illyricum. Here are the two parental species and their hybrid, even at this distance quite evidently morphologically intermediate. Whole fronds of the parents and then the hybrid, Polysticum illyricum. Detail of the two parental species and their hybrid, once again showing it to be intermediate in character. No wonder they hybridize. Now, what about this hybrid forming colonies? In 2008 and 9, I accompanied the a.s.l Field Club when they met at this West Highland site by Stroan Cruvy, not far from mountain botanists' favourite place to visit, Inch na Dampf. The habitat is blocky scree on a steep slope below limestone crags. It's an ankle-twisting habitat. Here we see two plants of Polysticum illyricum and one of its parents, Polysticum lonchitis. The late Ken Truran of the British Pteridological Society examines a plant, but when we look closely we see he is among many. Another BPS pteridologist, Roger Golding, in a similar adjacent situation. So many hybrids growing together is most unusual, but there is a fascinating explanation. In fact, there may be two explanations, but one turns out to be more likely than the other. In a 1972 paper, Anne Sleep, whose 1960s PhD project included a comprehensive review of the genus Polysticum, observed, P. illyricum is the commonest Polysticum hybrid occurring on the continent and is often found, sometimes in profusion, in the mountainous regions of Central Europe. So our puzzling ascent observation is not inexplicable after all. In the lab, Sleep discovered that 2 out of 3 Polysticum hybrids are unusual in that they are not completely sterile. So next, the third hybrid, the really rare one, is the sterile odd one out. A more detailed discussion of all hybrids will follow description of this third hybrid, Polysticum lonchitiforme. Polysticum hybrid lonchitiforme, P. lonchitis crossed with P. sativum. In Britain and Ireland, Polysticum lonchitiforme has been found in only one place, several plants on limestone around the borders of counties Sligo and Leitrim. So back to the 3 hybrid diagram, and on to the extremely rare diploid hybrid, Polysticum lonchitiforme. Even worldwide there are records from only 3 places, in Ireland, Hungary and Greece. Whole fronds of the parental species, and then with the hybrid Polysticum lonchitiforme. Although during her PhD research, her thesis presented in 1966, Anne Sleep could only perceive that Polysticum lonchitiforme was a hybrid that ought to be found in the field. She knew and understood it well enough once she had bred it from its parents under laboratory conditions, and many years later, in 1977, when she visited the Irish site, she at last saw it growing in the wild. Then she returned to Leeds, where she confirmed her earlier research discoveries. One of the 4 plants student Anne Sleep synthesized during the 1960s. The larger of 2 fronds she illustrated as silhouettes can clearly be seen to be morphologically intermediate between the 2 parents. However, some of the few botanists who have described P. lonchitiforme had difficulty distinguishing it from P. illyricum. We will first consider each of the 3 localities separately. The earliest found was reported by Eugen von HALÁSCY in 1904. The plant was collected in 1898 in the Taotas Mountains of southern Greece. The location is clearly provided online by the JECQ virtual herbarium on the mountain called Prophytis Ilias. The single specimen was collected by Karl Hermann Zahn, later documented by von HALÁSCY. The specimen was deposited in a Greek herbarium. I was pleased to find an image of it on the internet. When I first examined this online image of von HALÁSCY's herbarium specimen, my first reaction was, that's P. illyricum. Then I discovered Anne Sleep's 1966 appraisal of it. The type material, however, appears to be very close to P. illyricum in its morphology. Further down the page she revealed that one parent of P. illyricum, P. aculeatum, is rare even in northern Greece and absent from the Peloponnesos, whereas P. cytiferum, a true parent of P. lonchitiforme, is common there. She therefore concluded, this information therefore suggests that a single record for P. lonchitiforme from the Taygetus Mountains may indeed be referred to the hybrid P. cytiferum times P. lonchitis. Was that the matter settled? Von HALÁSCY in fact received Zahn's specimen via an intermediate botanist, Theodor Heinrich Hermann von Heldreich. It's not inconceivable that during the transfer, the original specimen of Polysticum lonchitiforme somehow got swapped with one of Polysticum illyricum, which was then incorrectly labelled lonchitiforme by von HALÁSCY. Gabor Vida, who discovered a Hungarian P. lonchitiforme in 1981, at first also thought it was P. illyricum. However, by that date, Manton and Sleep's technology was available to apply to it, and then he was surprised to find that it was in fact P. lonchitiforme. To quote, during meiosis, about 40 univalents and 21 bivalents were found. Accordingly, the hybrid is a diploid combination of P. lonchitis and P. cytiferum. No such confirmation was possible for the Greek specimen, so my third hypothesis might just be worth considering. I went back online to see if any more information was available. I reloaded the herbarium sheet. No matter how much I had enlarged the image I'd downloaded, resolution remained poor and critical analysis impossible. However, when I zoomed in on the original, resolution increased until I got a very reasonable image, which I processed with Photoshop. Allowing for the fact that this is an 120-year-old dried pressed frond, it's quite clear that it's not Polysticum illyricum after all. Let's compare fresh and herbarium specimens. In life, P. illyricum is, like its parents, tough and leathery, a condition that is retained when it's dried and mounted. In life, P. lonchitiforme displays its P. cytiferum genes, its frond texture being relatively thin and soft, which is evident in the Greek specimen. I've changed my mind and propose that the von HALÁSCY specimen better fits its original identification as Polysticum lonchitiforme. The Zahn von HALÁSCY plant may well have been unique, but they collected and killed it. A fully reliable identification has come from a site in Hungary. We have a relatively modern assessment of P. lonchitiforme, published in 1981 by Manton's student and colleague Gabor Vida. He found the hybrid growing in woodland between the Danube and Budapest and applied Manton's methodology to it. The wild plant provided excellent chromosomes and a confident identification as P. lonchitiforme. We can see that the Hungarian frond is, at first sight, very similar to the Greek frond. Until he tested his plant, Vida thought it was P. illyricum, but was able to check his hybrid's chromosomes and found them to be those of a diploid, not a triploid, refuting his initial identification and confirming P. lonchitiforme. Our third site for P. lonchitiforme has been visited frequently, indeed in recent years. The adult plants, of which there are several, do not resemble P. illyricum and appear morphologically intermediate between the two proposed parents, P. lonchitis and P. cytiferum. Early in this study I had little information or material to look at. Anne Sleep's 1960s research took place before she ever saw P. lonchitiforme in the wild, and all her understanding of the hybrid, reported in her thesis, was provided by 4 plants she synthesized in the lab. As far as publications go, it seems she didn't continue her research after seeing wild plants for herself in 1977, not very surprising because she suffered from systemic sclerosis for over 20 years and was already quite ill at the time. Therefore, we have no cytogenetical studies of field-collected material other than Vida's Hungarian plant, but fortunately that agrees with Sleep's pioneering work, so we do have a complete picture. The Irish records come from visits by local botanist Maura Scanell in 1974 and again in 1977, when she was accompanied by Anne Sleep herself. Then in 1984 a joint British-Irish group, also led by Maura Scanell with Con Breen, provided confirmation. There are now 10 specimens in the Dublin herbarium, 2 of which I describe below. Within the past decade or so, others have visited the site, and several plants have been found, all within the same general locality. Thanks to the generosity of pteridologists who have visited recently, the late Ken Truran and Roger Golding, I have some photos of the Irish P. lonchitiforme, and it doesn't resemble P. illyricum at all. As this presentation was approaching completion, I received photos of herbarium specimens of Irish P. lonchitiforme, and they really don't resemble P. illyricum. The best preserved was collected in 1984. It's a very large frond, pressed while it was fresh and green, an excellent preparation. When examined closely, the pinnules can be seen to be stalked, as in P. certiferum, though they become fused to the costa towards the pinna apices. This is the most pinnatifid specimen I've seen, but it is the best preserved and the most mature among the few that I've been able to look at. Here, from 1974, even though they're past their best, we have a fascinating set of 4 fronds, but one looks completely out of place. The light brown frond looks completely different from the other 3, and it's hard to accept that it came from the same plant. It looks very like a frond of Polysticum illyricum has been included with the 3 P. lonchitiforme fronds, so we'll focus on the most informative of those and overlook the outsider for the time being. The best of the 3 probable P. lonchitiforme fronds on this sheet, like other convincing specimens, has a good number of distinct pinnules that towards the pinna apex gradually fuse with the costa. Again, they're quite different from the herbarium specimens of P. illyricum, as well as the living specimens I've seen for myself. This plant was delivered to Anne Sleep, and as Vida reported, she proved it to be Polysticum lonchitiforme. If we return to the photographic comparison of the 2 hybrids, the differences are clear, and one feature stands out that would affect the preservation of pressed fronds. Like those of its parents, P. aculeatum and P. lonchitis, P. illyricum fronds are tough and leathery. In contrast, P. lonchitiforme fronds betray their P. certiferum genes. They're soft, both visually and to the touch, not at all leathery. That should be reflected in herbarium specimens, in that P. lonchitiforme fronds will shrivel more than those of P. illyricum, and that seems to be the case with the Irish specimens. Frond texture is not a diagnostic character, but what we and ornithologists call GIS, first impression, is usually helpful when combined with experience. If we could view specimens of both hybrids, side by side, fresh and herbarium, summer green and winter brown, we should probably be able to tell them apart initially by their manner and degree of shrivelling, then all their other differentiating characters. I was idly scanning the internet to see if I could find any more lonchitiformes, and to my astonishment I found one, another rare herbarium specimen, this time in the keeping of the Upper Austrian Museum for Regional Culture. It absolutely is Polysticum lonchitiforme, but the label lacks the most important information about it, where it originally came from. Unlike 2 other herbarium sheets I found at the same time, this one has the correct name. Those 2 other hopefuls from France and Italy, labelled P. lonchitiforme, are quite obviously P. certiferum. The label of the correctly identified lonchitiforme also tells us that the plant from which the frond came was in cultivation, not in the wild, and its location was the garden collection of D.R. Peters in the town of Söderbrarup in Schleswig-Holstein, northern Germany. It's dated 12th of May 2009 and was deposited in the herbarium by W. Limburger. Its identity was authenticated by Squiggle. I managed to locate Wilfried Limburger in Ottenheim, Austria, wrote to him, and he directed me to D.R. Peters, who turned out to be a well-known pteridologist, Dr. Bernd Peters, who indeed had a fern collection in Schleswig-Holstein. The published records for the hybrid's parents strongly suggest that the site of collection, other than Dr. Peters' garden, was not Schleswig-Holstein. So next I wrote to Dr. Peters, and his reply directed me to another German who gave him the plant, he having received it from Nobel Prize winner Professor Tadeus Reichstein, sometime a colleague of Manton Sleep et al. In 1977, Reichstein attended a British Pteridological Society excursion to Ireland. They visited the P. lonchitiforme site at Glenade and found several plants. I find it difficult not to speculate that Reichstein collected a plant and took it back to Germany. In those days, digging up a coveted plant wasn't taboo as it is today. The frond itself remains fascinating, and its similarity to the long-lost Greek specimen is undeniable. If only we could find out where it came from. I hope soon to find out if my speculation is correct. And it's not finished yet. Again, idly Googling, I found the details, unfortunately not an image, of a herbarium specimen from northern Spain. I've written to the University of Leon and hope to see it soon. Lonchitiforme is quite likely because both parents are present and common all along the top of Spain. A reasonable time has passed and I have not received a reply from the Leon University Herbarium. Here is a summary of what I found out. All the accession data are provided at the Hybrid's Global Biodiversity Information Facilities occurrence page via the link above. Maybe somebody else will be able to take this further. If correctly identified, this will be a very significant record, especially if it's still growing where it was found. Unfortunately, the convention has been to collect plants so they survive until discovered elsewhere at only one site. Check the notes below for updates. So, after waiting a reasonable few weeks with no replies from Spain or Germany, I must publish this video with two big questions remaining unanswered. 1. Where did the Schleswig-Holstein specimen originate, and am I right that it was collected by Reichstein in Ireland? 2. Is the Spanish specimen correctly identified, and what is known about it? Oh yes, there is one more. Will Polysticum lonchitiforme ever be refound at that old Greek site, perhaps by some energetic young pteridologist who must be encouraged? The arrival of a new species. Polysticum aculeatum probably evolved from a rare chromosome doubling event in P. lonchitiforme. The tetraploid was fertile and became very successful. Manton's and Sleep's research showed that it is likely that in the past a hybridisation event occurred that created our now common species Polysticum aculeatum, the hard shield fern. This is where the significance of that species being tetraploid enters the story. Instead of remaining a potentially sterile diploid hybrid, offspring were produced that had lonchitiforme's chromosome complement doubled, a not infrequent occurrence in plants called autopolyploidy, rendering the new offspring fertile. It's been inferred that proliferated and became a common new species, while when formed, the conventional diploid hybrid remained a rare occurrence. Sleep began investigating this proposed origin of a new tetraploid species. Numerous attempts were made to cross P. lonchitis with P. citiferum, and several sterile diploid hybrids were successfully obtained. When mature, fertile fronds were treated with the spindle-arresting substance colchicine in an attempt to induce chromosome doubling in the dividing mother cells. However, it will be some time before it is known if the successful resynthesis of P. aculeatum has been achieved. It was achieved. Although she had not yet finished the experiment in time for her thesis, as Vida reported, she did succeed. Sleep successfully crossed P. citiferum with P. lonchitis, and the few progenies of the mostly sterile diploid hybrid, synthetic P. lonchitiforme, were fertile tetraploids, presumed synthetic P. aculeatum. Job done. That concludes our exploration of the Polysticum triangle. There are many more Polysticum species beyond the British Isles, some of which are probably implicated in the ancestry of ours, just as we've been able to predict the evolution of the tetraploid Polysticum aculeatum from the experiments of the late Sleep. I'll now do my best to explain the cytogenetics of our Polysticums. 42 may be the meaning of life, the universe and everything, according to Douglas Adams, but the genus Polysticum has to be the exception. The number 41 is the foundation of the genetics of these ferns. By now you will have noted that the chromosome numbers of all Polysticums named in this study, and indeed all Polysticums, are divisible by 41. Their base monoploid chromosome complement is 41. 41 is crucial in the way Polysticums interbreed. That's the number of chromosomes in the genetic haploid condition of reproductive cells. This discussion will rely heavily on Anne Sleep's 1966 PhD thesis, which dealt in detail with the cytogenetics of the genera Asplenium and Polysticum, the latter of which we are examining here. Here are some important extracts from the text which particularly concern us. A phenomenon completely new to the ferns is the occurrence of widespread pairing at all levels of ploidy, which has been found in hybrids involving the species of Polysticum. The unusual frequency of occurrence of 2 of the 3 British hybrids is explained by these observations, that during meiosis, the cell division mechanism that produces the spores, the chromosomes segregate neatly into regular 41s, conferring a degree of fertility among the spores produced. Sleep continues, it has invariably been found in all the diploid hybrids investigated, regardless of seemingly gross morphological differences between the parental species and also some triploid and tetraploid hybrids. Therefore, our triploid hybrid Polysticum lonchitiforme does not have these neat chromosome pairing properties and is most likely sterile. We can't be certain because it is extremely rare and wild plants have received insufficient analytical attention. It's important to note that we humans perceive what Sleep described as gross morphological differences between the Polysticum species. We think they all look different, something of which their chromosomes seem to be unaware. In spite of differences we see, by which we automatically predict sterility, semi-fertile hybrids are a probability. Only when the number of paired chromosomes approximates to the monoploid number, in this case 41, can homologous pairing be safely inferred. It's that regular number 41 among meiosis-pairing chromosomes that confers the fertility. The occurrence of such widespread chromosome pairing in a large number of Polysticum hybrids between supposedly unrelated species is a phenomenon not previously encountered in the vascular plant division Pteridophyta. In the British and Irish flora, that is true of the hybrids Polysticum bicknellii and Polysticum illyricum, which for hybrids occur relatively frequently and plentifully, but it is not the case with the rare and almost certainly sterile Polysticum lonchitiforme. We can see it happening in microscope preparations of individual cells during metaphase 1 of meiosis. For the method used by Sleep, refer to Manton's 1950 book Problems of Cytology and Evolution in the Pteridophyta, without which no pteridologist could function pteridologically. Because Sleep didn't illustrate the metaphase 1 chromosomes of the British species, I resorted at first to Manton's book. Now, meiosis, which allows cytogeneticists to ascertain the numbers and sets of chromosomes and their impacts on reproduction. When the cell division mechanism meiosis is just beginning, the stage known as prophase, the nucleus disintegrates and the chromosomes condense, becoming visible under the microscope of magnifications around 1,000 times. They become attached to a structure consisting of microtubules, the spindle. They pair according to their genetic compatibilities and form a disc across the middle of the cell, shown here in side view, the metaphase stage that is of interest to cytogeneticists who need to view the disc at 90 degrees to the way it's shown here. They know how that's done. The chromosome pairs are drawn apart by the spindle, separating the individuals that have paired as bivalents. Under normal circumstances, that's pretty well all of them, but in hybrids things are usually very different. Once separated, the 2 chromosome sets are bound in new nuclei, and the cell divides into 2 daughter cells. After a second division not shown here, the result is 4 haploid reproductive cells which will become spores. Here we see a couple of Manton's FERN metaphase preparations, indicating just how difficult interpretation is going to be. FERN chromosomes are much more difficult to work with than those of many other organisms. Side by side, what the microscope sees left and Manton's explanation of it right. This is a relatively easy one. And now a tricky one, in which it was necessary to distinguish successfully paired chromosomes, bivalents, from unpaired univalents during metaphase in meiosis. Bivalents are successfully paired chromosomes, above. When a complete set pairs with another complete set of compatible chromosomes, they form bivalents and fertility will result. However, in hybrids, some or many chromosomes may fail to find a matching chromosome and remain univalents. When there are insufficient bivalents and numerous univalents, spore production fails. I could find little by way of rationalisation or discussion of Manton's illustrations of Polysticum chromosome behaviour and eventually asked Google AI for assistance. It provided surprisingly plausible explanations, which I'm confident were pretty well correct, so I was obliged to conclude that it evidently had more information sources I couldn't find, and that Manton was able to deduce more from her specimens than her published works provide. Chromosome behaviour of the species. Google AI said, in sexual Polysticum lonchitis, holly fern, univalents are typically absent during metaphase 1 of meiosis because it is a regular diploid species. It is fertile. I'll leave this example up for a little longer than others to allow you to register the standard format of this and the following slides. Google AI. In Polysticum cytiferum, the soft shield fern, univalents are typically absent during normal meiosis, as the species is a sexual diploid with regular chromosome pairing. It is fertile. As already discussed, Polysticum aculeatum probably resulted from chromosome doubling in the hybrid P. lonchitiforme. Google AI says, in Polysticum aculeatum, hard shield fern, a fertile allotetraploid, 2n equals 164, univalents are typically absent during normal meiosis. You will find 82 bivalents at metaphase 1, which is the hallmark of its stable chromosomal pairing as an established species. That's all very well for him to say. By simple counting of the chromosomes in Manton's explanatory diagram, in which some black blobs are attached or overlie others, I have been unable to confirm exactly 82 bivalents, and there should be no univalents, but she seems to show 8. For an established tetraploid species, you might expect 82 bivalents and no univalents. If Manton's diagram is true, the small number of univalents here presumably do not interfere significantly in fertility, for the species is undeniably fertile. Chromosome behaviour of the 3 hybrids. During meiosis, the 123 chromosomes of the triploid hybrids, Polysticum bicknellii and Polysticum illyricum, routinely segregate into 2 groups of 41 bivalents and 41 univalents. The probability of viable spore production is greater than in most hybrids. With a full set of bivalents, the univalents are less likely to cause sterility. Both of these Polysticum hybrids are at least partially fertile, as their reproductive capacity in the field implies. At meiosis, the chromosomes of the diploid hybrid Polysticum lonchitiforme segregate irregularly and variably. In the cell illustrated here, 70 univalents and 6 bivalents, but as you can see from Anne Sleep's cell counts, outlined in red below left, meiosis in this hybrid is a mess. In this meiosis, pairing is highly irregular, fertility will have been inhibited, and the production of viable spores was at best highly unlikely. We do have the good fortune to have a relatively modern assessment of P. lonchitiforme in 1981 by Manton's Hungarian colleague Gabor Vida. He found the hybrid growing in woodland not far south of the Danube in Hungary and applied Manton's methodology to it. The wild plant provided excellent chromosomes. However, Vida's initial identification before chromosome counting was P. illyricum, but his careful cytogenetical analysis proved otherwise. True P. lonchitiforme. Vida wrote, the mitotic chromosome count, however, indicated a diploid level, 2n equals 82 chromosomes. Remember that P. illyricum, which he thought he had there, is a triploid with 123 chromosomes. During meiosis, about 40 univalents and 21 bivalents were found, implying hybrid status. Accordingly, the hybrid is a diploid combination of P. lonchitis and P. cytiferum, Polysticum lonchitiforme. In spring, the behaviour of unrolling fronds is characteristic of each fern genus. A few examples. Shield fern croziers fling back their heads, while those of male ferns just unroll and then drop. Oreopteris croziers are hilarious.